This is where this was something I was discussing with Sahel after the class as a doubt. Anyways, where did we start in the last class? Here, right?
We did selectable markers.
Selectable markers. Yes, correct. So your voice was lagging. Let me try switching off my video and see. Can someone speak something? Just as a test. Just say hello, someone. Am I audible, people?
Hello, sir.
Yes. Yes. Okay. Great. Cool. Okay. So who would like to give a quick summary today? I think someone should volunteer today. Abia, how about you giving a summary? I very frequently go to Sahel, Matthew, and Fatima. Abia, can you speak? It will be a good opportunity if you give a quick summary of the last class. Are you there? I don't know if Abia can communicate with me. Okay, then we're left with Fatima, Matthew, and Sahel. Anyone? Because it's difficult to pick between you three. So anyone who would like to just volunteer? Okay, Abia's mic is not working. It's okay, Abia, no issues. Please get your mic fixed, because otherwise you will miss all the opportunities to just give a summary. That's the only thing. Okay, others, a quick summary. But you're waiting for me to call someone's name. Because I know. Yeah, okay. Because I know whosoever's name I'll take will say yes, sir. Okay. So it's better you do. Yes, Sahel, thank you so much. Let's go.
Yeah, so last class we started off by discussing the start of the chapter biotechnology. Then we came into how a recombinant DNA is made, which is for it, the first thing is we need a plasmid vector, or also called cloning vector.
Yes.
And then we discuss the characteristics of a plasmid vector. And the first thing it has is the origin of replication, which controls the copy number. And also it's called.
It also controls the copy number, but its main function is it also, like, it has two. It serves two important purposes. The second is that it controls the copy number. What is the most important thing? Why we are putting it?
It's where
it's the name. Yeah.
Yeah, it's the origin of replication, where it happens. Actually, it starts the transformation, right?
Yes. So it's actually origin of replication, as the name suggests. It is the origin of replication, and all we want is our vector to go inside the host and replicate, make more and more copies of our gene of interest that we have inserted. So if our vector does not have an origin of replication, then it will not replicate. Correct? Yeah.
Then we have selectable marker, which distinguishes the transformants, and then we have antibiotic resistance gene. It is an antibiotic resistant gene, and it helps. Then it has transformants, which combination of vector plus resistance.
No, basically, what does it do? Selectable marker. This is again another sequence of the DNA that helps us to distinguish or differentiate or identify the transformants from the non-transformants. So most of the times the selectable marker is an antibiotic resistance gene. It can also be something else. I gave you a homework to read about insertional inactivation. So that is another way of having a selectable marker instead of.
It helps in identifying and eliminating the non-transformant
Yes. So yes. So
right?
when we will identify transformants, then only we can eliminate the non-transformant. Correct. So that's what selectable marker is for. Yeah.
In turn,
Continue.
helping in growing of the transformant, right?
Yes. Right.
And we talked about transformants. So it's a process in which a piece of DNA is introduced in a bacterium, host bacterium, right?
Sorry, come again.
Transformation, we discussed about transformation, right? Which is a process in which a piece of DNA is introduced in a host bacterium.
Yes. So that is transformants. We have not discussed how to make transformant, because that includes competent cells making. So that I will teach you today. This I just told you that selectable marker is used so that when we successfully we have made transformants, then we need to identify which are, for example, here the red ones are transformants. So they can be isolated and then they can be cultured further. Transformants are those who have our recombinant DNA or the vector, right?
Right, sir.
Yeah.
Yeah, sorry for the disturbance.
No, it's fine. It's okay. We all are at home, so
Yeah. Then
yeah.
that's it. I guess you can scroll it down.
Yes, correct.
Yeah. Then we talked about cloning sites, and it is where the foreign DNA is linked, and the vector has. I mean, like, in order, it's like foreign DNA is linked, and it is also the site recognized by the restriction enzymes to create a so what is it there? Cost.
To
What did you
create
write
a
here?
cut.
Cut. Okay. Cut in the
Yes,
DNA. Right.
I have a very bad handwriting, but
No,
I
it's fine,
realize
sir.
it becomes even bad when I get conscious about it. Yeah, cut. Come on. Yes.
So what it means is actually, like, in order to link an alien DNA in a vector, so the vector needs to have very few and preferably single recognition sites. Means like we have to recognize where
Yes.
we have to actually attach the alien DNA, right?
Yes. Correct.
Yeah. And this is then used by the restriction enzyme.
This is not used by. So restriction enzyme is the protein molecule that creates that cut. So there can be different types of restriction enzymes, exonucleases, endonucleases, and the one that creates the cut, they are called nucleases. If they create the cut anywhere in the DNA, they are called endonucleases. But if they are degrading the DNA from the sides, that's called exonucleases. Correct? And I just discussed that how it was discovered. Yeah.
That part I don't remember, like the discovery part.
Anyone would like to help Sahel? Arpit has joined. Hello, Arpit.
Good evening, sir.
Good evening. Would you like to give a quick summary for the
Actually,
restriction
I'm
enzymes
having
part?
my food right now, so can I?
Okay. Yeah, okay, great.
But
Okay,
actually,
so
I had
whenever
gone
of
out
you
somewhere, so I had not taken my
have you
lunch. So
cool. I can understand. That's the life in PhD. That's the regular life in PhD. Having food in conferences. Sometimes I also eat during the class. Yeah. Okay. Matthew or okay, Kulsum just joined, but she's not here. Matthew, remember the restriction enzyme part? See, you have to go back and revise, because it does not matter how good. So you may be wowed, or you may feel that, oh, Rahul explains really good, so we understood everything. My explanation is just limited to the class. When you are out of that class, when you shut down that Zoom meeting, after that it's all you. I can only take as much space in your brain as much you allow me to. So, and for that you have to revise what I explained. because I'm 100% sure that yesterday when we were discussing restriction enzymes, Sahel understood everything because all of you are very sharp in understanding. But if you cannot explain it the next day, that tells that you are procrastinating, which, you know, if even I do a lot, but when it comes to some concepts, you have to be more responsible, especially at this age where you have a lot of things on stake, your marks, your exams, your future career directions, etc., etc. So no pressures, but just enjoy the process, but please do revise. These are interesting things, but simultaneously these are complex things, complicated things, not very easy to just
Sir, I think I
yeah.
remember. I just went
Now
through
you
the
remember
notes.
now?
Yeah, I just went
Okay,
through the
you just
notes.
needed a pep talk. Oh, that's
Yeah.
great.
Yeah. So
Cool. So yeah.
So restriction enzymes, as you said, there are endonucleases, exonucleases, and how it was actually discovered was there was an experiment on E. coli where we found that E. coli could restrict the bacteriophage which attacks the E. coli, and it could restrict the infection and growth of it. And because of two enzymes, and because of this, two enzymes were isolated, which could actually restrict it. And the first
Yeah.
enzyme, we added a methyl group to the viral DNA, and in the second one we cut the DNA into
Not
fragments.
we did not add it. So the first enzyme was adding methyl group to the viral DNA, and that's how it was. This was the mechanism by which it was restricting the growth. So this enzyme we were not interested in because it's not a nuclease. It is just silencing the DNA, okay, not breaking it or degrading it. The second enzyme was cutting the DNA into fragments. Now that is something interesting to us. Okay, cool. There is
Sir,
something that can cut the DNA. Yes, Arpit.
sir, I have a doubt. Sir, this methyl group, sir, this does not allow the restriction endonuclease to cut the DNA. Is it like that?
No, this is not restriction. So this enzyme 1, adding methyl group to a DNA silences the DNA. Silences the DNA means it does not allow the the replication machinery to come and bind to the DNA and start the replication. Correct? It's like hiding the DNA under a blanket. So the restriction enzymes won't be able to, the replication enzymes won't be able to find it and won't be able to replicate it or transcribe it or anything. So the excess is cut. It's like you just hide the DNA with methyl groups.
Sir, this group hide the sequence.
It just binds to the DNA. So point is there is a rope. You have to hold on to a rope, okay?
Yes, sir.
But if I just bind balls to that rope, a lot of big balls everywhere on the rope, you won't be able to hold to the rope, correct?
Yes.
So that's DNA. The rope is DNA. The rope should be accessible so that something can hold on to that rope. But these methyl groups are like balls, if you understand. So let's say, let me try to see if I can make you understand. So let's say this is a double, my bad. Let's say this is a double-stranded DNA, correct?
Yes.
These are the bases. And let me just tell you specifically, you don't have to remember it. So it has ATGC, C is cytosine. So cytosine can be easily methylated. So you can add a CH3 group, which is a methyl group, to a cytosine base easily. So now this is accessible, right? Now imagine these red balls are CH3. So I added a big CH3 here, then another C was here. I added a CH3, I added a CH3. If there is another C, I added a CH3. So I just keep adding these CH3 groups to DNA wherever cytosine is, correct?
Yes, sir.
You understood now?
Yes.
Now imagine this in 3D. Now this black thing is the enzyme that wants to come and sit on the DNA and access this region and start replication. Will it be able to come and sit?
No.
Because there is a steric hindrance now. So that's that way DNA becomes inaccessible for replication. It also becomes inaccessible for transcription as well. So that was what enzyme 1 was doing. So it was not like it was not a nuclease enzyme, but a methylase, methyl transferase to be very specific. It's called DNA methyl transferase enzyme. So enzyme 1 happened to be a DNA methyl transferase. It adds methyl group to the DNA. Enzyme 2 was cutting the DNA and hence it was restricting the infection. So that is an endonuclease because it's cutting the DNA. So this is what we were interested in. Is that clear, everyone?
Yes, I got it.
Perfect. Yes, Sahel, over to you.
Yeah, then we discussed about the different examples of restriction enzymes. ECORI1 was the first restriction enzyme to be isolated and found out. And
It's
then we
eco
Eco
discussed
R1.
about,
right?
Yeah, so Eco R1.
yeah, ECOR1. And then we discussed about the naming of it. Yeah. So for example, hint 2, HIN stands for Haemophilus influenza, while D stands for the strain bacteria, and then 2 stands for the order of isolation. This is the general naming sequence. And same for
Correct.
ECO R1. The first eco part is the generic name or for the bacteria, Escherichia coli. And R is
Yes.
for the strain, which is RYS. And 1 is the first because it's the first one to be isolated. And then some of the examples are CLAV1, then BAMH1, then SAL1.
Yes, and I ask you to go back and just as a exercise,
Yeah.
try to find there. So anyone did for BAMH1 or all these three especially? Anyone? Let's start with BAMH1. It's in your. So what is BAM? Of course, BAM must be the name of the source, right? So this is the name of the source. This is the
Yes,
name
sir.
of the
It's there in the
strain.
PBR 322 plasmid.
Yeah, so
Sorry,
PBR 322 is the most used vector, most used
Yeah.
plasmid as a cloning vector, and you can put anything in PBR 322. You know, all these restriction sites are available. So nowadays, commercially, you can buy plasmids where all these sites are available. You just have to choose a restriction enzyme for your experiment. It will go and cut because all these plasmids are genetically engineered and are available for research purposes. So you don't have to from scratch make plasmids these days. You can just order them, or companies will make for them if you want any modification. Okay. So in your NCRT, I think there is a diagram where they have shown a PBR 322 plasmid with all
Yeah, sir,
these
there
sites,
is.
right?
Yeah,
So
sir.
it just tells that you can have all these sites. You just need whichever site you want to cut at, you just use that restriction enzyme. Okay. BAMH1, anyone knows?
So
So
it's
BAM
like
stands for
tetracycline resistant, right?
if it is like resistance, yes. So no, we are not talking about resistance. So that is like what resistance you will. I'm asking what does BAM H1 stands for? Like HI and HIN stands for Haemophilus influenza. The name of this plasmid, where does it come from? So BAM must be the source, right? BAM. So it's
Yes.
Bacillus amyloliquefaciens. Amyloliquefaciens. You don't have to remember it though, but just know. Then H, you know, is the strain, right? So that strain is strain H. It's known as strain H only, and 1 is the order of. So this was the first restriction endonuclease that was isolated from the H strain of Bacillus amyloliquefaciens bacteria. Similarly, you can find for CLA and SAL1. Okay. I also told you to go and read about Agrobacterium tumefaciens. We'll be using it in the next chapter. Okay, so for the time being, let's come to the binding sequence. Yes, Sahel.
Then we discussed about palindromes. Means we can read it the same way from either side. Example, Malayalam, Nathan, rotator. Nothing much to discuss about that. And then we discussed.
No, but what was palindrome?
Palindrome is a sequence. No, if in terms of biology.
Why we started discussing about palindromic sequence?
Yeah,
There's a lot to
because
discuss about it.
Restriction endonuclease binds to it.
What is
it's
what?
the sequence in which the nucleotide binds, right?
This is the nucleotide. CTAA. Nucleotide is the.
Yeah, because example here CTT AA will bind to AATTC because in either ways it reads the same way, right?
Yeah, that's right. But how is it related to restriction endonucleases? What is its relation with the restriction endonucleases?
Okay, so restriction enzymes actually identify the palindromic sequence.
Yes, restriction enzymes always identify some or the other palindromic sequences, and they only can make a cut at palindromic sequences in the DNA.
So only at
and they don't make a cut exactly at the center.
Right.
Yeah.
Recombinant. Yeah.
Yeah,
So
they make cuts away from the center in a way that sticky ends are produced.
right, sir. And the sticky ends attach them, right?
These sticky ends attach to each other, and the sticky ends attach to the piece of our gene of interest if we have used the same restriction enzyme to cut both. And that's always necessary. If you're doing biotechnological experiments, you have to use the same restriction enzyme to cut your gene of interest and to also cut the plasmid vector, so that the same sticky ends are left in both these pieces and they can be ligated together easily to make a recombinant vector. Correct?
Yes, sir. Got it.
Everyone? And then I asked the question that how will we get to know, how will we isolate our gene of interest from that whole genome, the source genome, from which we want to cut a piece of DNA, which is our gene of interest. So how to isolate it, how to get to know it. So there's a technique called gel electrophoresis. And Fatima was discussing something about chromatograph in the last class, and we realized that why we cannot use that, right? We understood why we cannot use that here. So here we have to use gel electrophoresis. Now the term gel tells us that there is some kind of a gelatinous medium. Correct? We are doing all of this on a gel. The word electro is for electricity. It includes electricity. Phoresis is separation. Okay, kind of like to separate. The idea is to use electricity on a gel medium to separate certain things. Now, what are we doing here? When we cut DNA by restriction endonucleases, it will result in fragments of DNA. But these fragments, there'll be multiple fragments. Out of these multiple fragments, we need to separate our gene of interest. Now, the good thing is we already must be knowing about our gene. We also must be knowing about its size. So how big it is, what is its molecular size. So it becomes easy for us to separate it through gel electrophoresis. Now, gel electrophoresis is a technique which includes a gel. So the gel that we make is made up of a polymer. We use a polymer known as, anyone knows which polymer we use to make this gel? Agarose. So it is also known as agarose gel electrophoresis. Okay. In general, it's known as agarose gel electrophoresis. Now, agarose is a polymer, biological polymer, which is extracted from seaweeds. These are small plants present in the sea. Seaweeds, extracted from seaweeds. Now this agarose, if it's like a polymer, so what we can do is look at these two pictures. So these are long threads, right? If I allow them to settle together, cool. So depending on how much polymer I'm using, let's say this is 1 concentration, 1x concentration. So these polymers set together and cross-link together, and they leave some gaps in between, right? Do you see that there are gaps here? it's like randomly, if I allow some particular concentration of polymer to set together, there will be some gaps, right? All these red areas I'm marking are the gaps, correct? Everyone understands? Yes, no?
Yes, sir.
Now let's say I increase the concentration of the polymer. I took so much. I took 5 times more concentration of the polymer for the same volume. Now what will happen? Again, these polymers will set and cross-link in the same volume, but this time the concentration of polymer is very high, 5 times high. So what will happen? Let's say this is 5 times higher volume. So what will happen? Look at the pore size. What happens to the pore size which is present in between? What happens to the pore size if I use high concentration of that polymer to make a gel? Where do you see the pore size is bigger?
In less, in 1x, that is. Yeah.
Yes. So I can modulate the pore size of the polymer of this gel based on the concentration of the polymer. This is not the best way to represent it, best diagram, but you understood the concept, right, everyone?
Yes, sir.
Yeah. So through this, we can use different concentrations of this agarose polymer, and we can let it settle in the form of a gel. So this is the gel which has been formed. So in 3D, it will look something like this. It's like a piece of cake. In that gel, we will make some wells, like some regions which will have indentations. This we can make by using plastic combs when this gel is setting. So when you boil it, this is liquid. When you allow it to settle, it solidifies into a gel. Now in these gels, we can load our sample. So let's say I loaded the sample A here, sample B here, sample C here, and sample D here. In the first well, we always load a marker, which is called a ladder, DNA ladder or marker. Now, why is that important? So these are wells. Now, let's say depending on what is the size of the DNA of our gene of interest that we want to separate, we will decide the concentration of agarose so that it has a particular pore size. And then all of this thing is kept in a liquid, which is called buffer. It's like kept in a box, which is full of liquid. And we will connect 2 electrodes to this. Now, all these samples are DNA, right? DNA samples, which are fragmented after restriction endonuclease treatment. And this ladder, I know, is of different sizes already. So this it's available that what will be the mixture of different DNA sizes present in this ladder. So this will act as a reference for us. Okay. Now what happens is DNA is negatively charged molecule, correct? Everyone?
Yes.
Yes, sir.
So we will add a circuit here. On this side will be positively charged terminal, and this side will be negatively, sorry. On this side will be negatively charged terminal, and on this side will be a positively charged end. Now DNA being negative charge, when I'll allow this electric current to pass, which direction should it move to? Due to the force
Positive.
towards the positive, right? Towards this direction. Now we already know that in the first ladder, we have put a reference marker. And the reference marker will already tell you that it will make 6 bands, 10 bands, whatever. So let's say it makes these 6 bands. And the size we already know. So tell me if this ladder is from 1 kb to 10 kb, let's say 1 kilobases to 10 kilobases is the size this reference ladder will tell us. So where will be 10 kb? Towards the negative or away from the negative? 10 kb means bigger DNA.
Towards the negative.
So bigger DNA will move slower, right? Through that pore, whatever is the pore size.
Yes, sir.
But smaller DNA will move very faster, correct? And it will go quickly away from the, it will cover the farther, it will cover the most distance in the same time that 10 kb will cover. So it will take half an hour for 10 kb to cover this much distance because there is a lot of resistance. The pores are very, very small, but the 1 kb can cover this much of distance because it can freely move through the small pores, correct?
Yes,
Then
sir.
this will be, let's say 8 kb, 6 kb, 4 kb, something like that, 2 kb, and then 1 kb. So now we know for our sample, we will see the same kind of, so whatever we know that our gene of interest is a 2 kb gene. So if I find in sample A a band here, which means this is a DNA fragment, which is 2 kb fragment or somewhere around it, right? I also find it in D. So if this is the pattern that I get, so which samples have my gene of interest? My gene of interest is a 2 kb gene. So which samples have my gene of interest? A
and D.
A and D, correct? So do you understand how did we isolate it?
Yes, sir.
Now what we will do is we will take a blade and cut out this part of the gel. We don't need this. We will cut from here, and we will actually we have to cut as minimal piece as we can, like this, so that we do not get any contamination, but we get all the DNA that we want. When we'll cut it out, then we can extract that DNA from that gel by a process called elution. By eluting out that DNA, it means you just again dissolve the gel and free all the DNA fragments that were present inside that gel, right? So you cut it, you do the process of elution, and after eluting, you get your DNA of that particular size. And then we can go for further processing. So this is how we will get our gene of interest from a whole genome, where we have used restriction enzyme to cut our gene of interest. Is that clear, everyone?
Yes, sir.
Yeah.
So,
So yes.
how are there two same gene of interest?
How are they two? They are two different samples. So in biology, whenever we do experiment, we don't just do it one time. So n equal to 1. Anything that is done in science just once is not taken as an evidence. Science has to be repeatable. If it is true science, how many number of times you do it, the results will come the same. Do you understand that? So the difference between a science protocol and any other protocol which is non-scientific, or where people have used intuition or faith or culture or their beliefs, is that any piece of document which is written on the basis of science, if you burn it, if you destroy it, if let's say that's not present on the face of earth, then if the new generation again wants to figure the reason out, let's say whatever we know about gravity, we just burn everything and no one from the next generation knows anything about gravity. But again, someone will start asking questions like Newton, Einstein, and Galileo, these people did, right? So when they will start asking the same questions and they will start doing the same basic experiments to find out the truth of nature, they will come to the same conclusion over and over again, because it's science. It can only be done one way, and you can come to the answer which is true. Any number of times you do that experiment, it will give you the same result. That's science. But if every time you're doing it and it's coming out to be different, that's not science, that's Chinese whisper. That's how different stories, cultures, faith have evolved, because every time you go to different places, there'll be different notions, different stories about different kind of phenomena that are happening. Do you understand?
Yes, sir.
Yeah. So that's why in science things have to be repeatable, things have to be reproducible. So this is sample A, B, C, and D. So we are trying to find, so sample A and D has our gene of interest, B and C does not have. So this A and D is basically taken from the same set of organism which has this gene. Then we use restriction endonuclease to cut that gene, and we can find that gene in both these samples, which is not present in this two. So there can be two gene of interests found because the sample is different, correct? And these are multiple, these are millions of DNA molecules. This is not just one DNA molecule of 2 kb, because DNA is very small, and if we have to look, it has, if it is visible, it is too many, so that we can see it as a band. So through elution, is that clear, everyone? This part is clear?
Yes, sir.
Yes, sir.
Okay, so through elution, the DNA of interest is extracted from the gel. Okay, so write down few points. Separation and isolation of DNA fragments. Separation and isolation of DNA fragments can be done through agarose gel electrophoresis. Through agarose gel electrophoresis. DNA being negatively charged molecule, DNA being negatively charged molecule can be separated by or you can just add fragments of the DNA. Fragments of the DNA being negatively charged molecules can be separated by applying an electric field through the agarose gel medium. Applying an electric field through agarose gel medium. Now this field, the voltage has to be very low, the current has to be low. It should not be very high intensity current that will just fry off every molecule. So we have to take care of that. It should be enough to create a pull or to create a push in the molecules due to an electric field, but it should not be strong enough to just destroy the molecules. So that thing can be standardized. Okay. Then write down the separated DNA fragments. The separated DNA fragments can be visualized. So how do we see these DNA bands? Because DNA does not have a color, right? So we don't see any color. This is just to explain you. On a gel, you can only visualize DNA if you stain the DNA. If you put a stain, like if you mark it somehow. So we stain the DNA with a compound called ethidium bromide. Okay.
So
Write down DNA.
which
Yes.
do we stain all of them?
Yes, we stain the whole sample, like everything. That's why we can see that different fragments are present. So DNA staining can be done. This is just to visualize the DNA where our strain is, and for how long do we want to run this gel. Because if you're running this gel, the DNA starts moving. Over time, you will see this last band going farther and farther. But you have to know where to stop, because if you can't see the last ladder, then it will just continue moving and come out of the gel into the liquid. It has happened to me also, where I have run the gel ulta. So I put the gel like this, but towards the well region, I by mistake connected the positive terminal. So what happened is the DNA just came out of the well and moved towards this opposite side and just fell into the jumped from the cliff into the solution, kind of. So I think it's a very classic mistake most of us have created. Most of us have.
So like it comes out of the well?
Yes, it can come out of the well because you're just pushing it. No, there's nothing stopping it to be just in the well. The only thing which is containing it is the well itself, is the gel itself. When it has come to the last part of the gel, you will just electric field just keep pushing it. So it will push it out of the gel because it has pores, and everything will come into the buffer and it's lost. So that's why staining, that's why staining and visualizing is very important. So DNA staining can be done using ethidium bromide. I think ethidium has a Y. No, it does not have a Y. Ethidium bromide. But we always have to be careful because ethidium bromide is a kind of carcinogenic to skin. It's a carcinogenic chemical, so we should not touch it with bare hands. So when we do this process, we always wear gloves and do not touch ethidium bromide by bare hands. If you touch it once, it's not like you will get cancer. It's just like you should not
someone
expose
must have
yourself.
touched it, right?
No, we can also test this into cells, right? Cell cultures. Some chemicals, their properties can be tested in the cell culture. If they create a tumor, or if we know that this molecule is capable of making the cell divide uncontrollably, it is a carcinogenic chemical, right?
Sir,
Like nicotine does. Yes.
Yeah.
sir,
So it's not like every poison was has to be given to some human, and if it dies, then if the human dies, then only we will say that it's a poisonous substance. We just know the biology, biochemistry of it, right? So in a snake venom, there are some chemicals. We know that it's a. I'll come to you, Fatima. Just 1 second. So let's say snake venom, there is a chemical. If I make an analog of it, which is something similar to it, I will be like, okay, this can also be venomous inside a cell. So let's check it inside a cell culture, a human cell culture, correct? Or in a mouse or in some other organism.
Yes, sir.
Yes, Fatima.
so if it is carcinogenic, so if you are putting it inside the DNA, can it not, change the genes and make the cell lose the means like this?
There is no cell here.
No, yeah, I know. But then this gene of interest will be going to a cell, right? To a plasmid, and then that will be going to a cell. So it will like eventually interact with a cell. And that ethidium bromide, you can't remove it afterwards.
You can remove it afterwards.
How can you remove
So it goes and binds. It goes and binds to the minor groove of DNA. So DNA has grooves. It's about its 3D structure. So this DNA does not goes and create some mutations in the DNA. If it does that, then it's not of any use. But if I just erase everything, just allow you to see that naked DNA. In the DNA, there are grooves. So sorry. Oh, this is what okay, let me show you here only. So some major portion. So let's say this part here is a groove. You can see these are grooves of DNA, where DNA takes a turn and it bends towards one side. So these are groove regions. So it goes and binds there. So when you are making vector, you make sure that this DNA is treated again, and this ethidium bromide is not bound to the DNA. Or when you visualize, after visualizing, you know that your DNA is there. Then you run on the gel just a sample, okay, to see that if there is a DNA. And when you elute it, so during elution, of course, we have to make. You're right. If it goes, if ethidium bromide goes inside the cell into large quantities, then it will start causing problem in the cell itself, into the host cell itself. because it affects the cell cycle, the cell division cycle. But we do make sure that when we are making a recombinant vector, it's not there. Okay, it's just for the visualization purpose on a gel so that we know that where to stop.
So afterwards, how do you remove it?
That's a slightly complex process, which is not scope of your syllabus. So we will discuss it after this part is done. Otherwise, it will take another 15 to 20 minutes for me to explain you the structure of the nucleotide and where it goes and binds and how can we remove it.
So have you ever performed this?
This is routine. This is routine. This you will, I think, if you do a BSc also, a BSc in biomedical sciences or biochemistry, right from there. So running a gel is a bread and butter of every molecular biology lab. So it just sounds very complex. Once you start doing it, you can just do it you can do it half asleep also. It's not a problem. So I have done it,
But then,
I don't know, uncountable number of times. Yes, Fatima.
Isn't it possible to have many failures in this? Means the experiment being failed.
All experiments in science are bound to failures even by just small problem. So there are thousands of things that can go wrong. Maybe when you are using the restriction enzyme that is contaminated with exonucleases, it's not just endonuclease. When you extract the DNA, there is something, even if you breathe or something from outside, even any contamination, a drop of sweat, even just a droplet, you sneeze and something goes inside it, it's gone, the sample is gone. Any contamination can happen in the first place. Second, when you are running it on the gel, you can see that while it's running on the gel, the DNA gets degraded and you are not getting any band. Instead, what you get in a degraded sample is a smear like this, which I have also received a few times. So your gel looks like this. This is the most frustrating thing any molecular biologist can see in his or her life. And this is called a DNA smear, that your DNA was degraded before or after you cut it through. So that experiment is failed. Other thing is what I did, you can just join ulta, different electrodes to different sides, and all your DNA can just jump off the cliff and go away.
Yeah.
Or else you used higher voltage than required, and your DNA, you ran it for 20 minutes. By the 20 minutes, the DNA is either out, or you ran it too less. You use the wrong ladder, in which case you will not be able to identify which band is your gene of interest. Anything can go wrong. So that's where scientists, they have to be responsible. You have to do it multiple times to know for sure that what you did is correct. It's not like you did something for once, you got something and you are like, okay, great. So for my own experiment, I had to run gels multiple times just to be sure that what I'm doing is correct. So in biology, minimum what is asked is n equal to 3. So you should have 3 replicates showing the same result, so that one can say that yes, your science is correct. Science, as I told you, science cannot work with friendships and just okay, he's my friend, so I should just trust him. No, whoever he is, you should trust the data, not the person. Okay, if the person is a scientist doing good and robust science, the person's data should talk for him or her. So science is based on criticism, a constructive, positive, data-oriented criticism. And a criticism in science is not about the person. So suppose I am doing an experiment and I'm presenting the results. So the person who's criticizing will not say, hey, you are a dumb person, what did you do? We'll say that, so I found some loopholes in your data. That has nothing to do with me personally. It's not a personal attack that, oh, he's suspicious of my data. I should not take it as personal attack. I should take it as a critique of my work. So I'll just talk to that person more and more. Yes, what do you think is the flaw? So I'll explain. If the flaw is correct, then I will get a constructive feedback. I will go make changes in my protocol, do it the more logical way, because I got to know one caveat that I was not aware of. And that is how science develops. So in science, you don't take criticisms in a negative way. It's a very positive thing. If no one will criticize your science, it will never grow. Correct? So there are many things that goes wrong in science. It has nothing to do with even all these brilliant scientists were wrong most of their life. They were just right a few times, and that changed the world. Newton was wrong thousands of times. He was right the one time, and the world got transformed. We started making rockets going to space. Einstein was wrong thousands of times, but he was right one time, and it changed the world. We understand how energy and mass can be converted. You understand? That's how it works. So being wrong in science, I think, should be celebrated, because if you're wrong, you realized what should not be done. So no one should repeat this. You realized one way. So I hope you all know about this one biographical story of Thomas Alva Edison. Edison was not the first person to create to invent bulb like we study in the books. That's a very simplified way to say who was the inventor of bulb. Thomas Alva Edison. Before Thomas Alva Edison, there were half a dozen of different kind of bulbs already available, like already invented and available. But the problem with all of these bulbs was they were very short-lived. What Thomas Alva Edison really achieved was making a filament that could keep glowing and keep giving out light and heat for a very, very long time. It was the most stable electric bulb that he created, that we currently use also. You know, the tungsten filament? Now, though, we are not even using Thomas Alva Edison's bulb. No one knows who invented LED, right? Who invented neon fluorescent tubes? Do you know who invented fluorescent tubes? So if you go to somewhere where they have written like hotel like this in tubes, so these tubes glow, that is not the bulb that Thomas Alva Edison created. He created this one, where there is a glass, then there is a space which is sealed with a system, a negative terminal, a positive terminal. Inside there is a tungsten filament like this, correct? You all have seen this? This bulb, remember?
Yes, sir.
Anyone? Do you use these bulbs in your house these days?
No.
I'm sure no one does, except for a few places where these bulbs are still used. So what happens here is when electricity goes in, this filament is made up of tungsten, and this tungsten is one of the most strongest metals, or there are other things also that can be used to make this filament. So this filament heats up, and when it heats up so much that it starts radiating, it becomes red and then fluorescent and starts radiating light out. Okay, that's how a bulb glows. So this was his achievement. But Thomas Alva Edison did not succeed in just one night. It took him years and years and sleepless nights, and he at least tried somewhere around 2,000 different ways to make a sustainable electric bulb, and he failed. So someone asked him that, you know, what did you like? Do you think that failing 2,000 times, how did it affect you? How do you feel about those 2,000 failed experiments? So he said that I feel good because, you know, I know 2,000 ways how to not make an electric bulb. Do you understand that, what I'm trying to say? So
Yes,
no one should be judged in science.
Yes,
something. Fatima.
Every time he learns something new.
Exactly. So no one is actually criticized, or no one is actually judged in science for being wrong or doing a failed experiment because you did an experiment that tells that you are a scientist. The same I expect that should happen in our education system in classes, that you should not be judged for giving wrong answers. You should be just judged for thinking. You are thinking, you know, your thought process can only evolve if you are not afraid of being wrong. If you're afraid of being wrong, then you will never be able to speak up until you are sure that you are right. And that is what education system is moving towards, which I do not agree with very much, because that way we will just become trained educated people. We will not become thinking brains or scientists. And we need thoughtful people more than ever in today's time, because the world is on the brink of collapsing, where some people like Elon Musk really thinks that we will go and terraform Mars and we'll start living there. But you know, that's just a very, very distant thought. My personal belief is it's always will be easier to preserve and protect Earth than to go to a planet that's dead and revive it from the dead. But I don't know. We should be more focused for our own planet than to be trying to revive Mars.
Is it not? I think it's not even possible. Or it is.
Nothing is impossible in science.
Yeah, it is.
We
It is
cannot say that it's
possible,
not.
but then like
Yeah.
keeping it on Earth is like more valuable.
Yeah.
See, what you're trying to do is you are trying to create Mars. You're trying to make Mars into Earth. But you have Earth. Why have you given up on Earth itself when it is alive and kicking? Protect it, preserve it, right? Make it more sustainable. Instead of thinking that, okay, this is anyways, okay, let's go to Mars. Let's go to Mars. There's no problem in that. Let's go to Mars. Let's go to other galaxies also. But let's not give up on Earth, right? That's what my point is. We should not. And I think we will not. Anyways, so yes, does that answer your question, Fatima, that anything can go wrong? This is whatever I teach you is the standard workable way. So this is the standard way that has been standardized and it works. So if I start teaching you what cannot work, then we will never be able to move from one topic. Okay? But just because you people are curious, it's a good thing. Okay, so this is how we can extract our gene of interest. Okay, through elution. So what after elution? I have two things now. Question is what next?
So to insert the gene of interest.
Perfect, Arpit. So we have our DNA of interest. We also have our plasmid vector. And we have treated both with the same restriction endonuclease. After that, we also have done electrophoresis and taken we have purified our gene of interest. So after that, we will make recombinant DNA using ligase. Using ligation, DNA ligation. So we will use DNA ligase enzyme. This is done. Now it is ready to be inserted into the host. Now, which is the what is the host organism? What is the host organism?
Bacteria.
Bacteria or yeast in some cases. So let's focus on bacteria for this discussion. So let's say I want to insert my recombinant plasmid vector containing my gene of interest into a bacteria, and then I want to amplify that gene. So how will I put it in bacteria, and why will bacteria take my packaged plasmid vector? I cannot talk to it, say that please do it for me, take some fructose as a reward. So cells normally, why will they take any piece of DNA from outside? And also we cannot just make DNA pass through the cell membrane. You know why? Because DNA is a hydrophilic molecule. What do you mean by hydrophilic? DNA loves water. Sorry?
Like you said, hydrophilic or phobic, sir?
Hydrophilic means
okay,
it loves water. So if it is hydrophilic, it will be lipophobic. If something loves water, it hates oil or lipids, correct? If something loves lipids, it hates water. That's how it works. And our cell membrane is made up of lipids. So if something is hydrophilic, it cannot pass through the cell membrane because cell membrane is lipid bilayer. So anything which is hydrophobic and lipophilic can only pass through cell membrane, correct? So DNA cannot easily pass through cell membrane. Now the problem is how to give this DNA to the bacteria. So we have to force it. What humans are really good at, when we cannot make nature work in a way we want, we force or we try to alter and change or challenge the nature. It has also yielded good results, better results. We have created medicines and all. We have helped humanity, saved the planet with doing that. We have also created a lot of problems. So here also we are trying to force the cells to take our plasmid vector. Now this forcing, we have given a very beautiful technical term to it, that we are making the bacteria competent to take up the DNA. So that's called competent cells. So now we have to, next thing is inserting plasmid into bacteria, which is our host cell here. And for this to happen, we have to make the bacterial cells competent. Okay. When I was doing this for my gene of interest, when we were trying to, clone the gene, making competent cells is very, what do you say, a responsible job. So you make competent cells in bulk, and then the whole lab will use it for many experiments to come. So we can make bacterial cells competent, and we can store it in -80 degrees where they metabolically become inactive and they are stored. They don't die. Of course, we store it in a solution that does not freezes at -80. So it should not be water-based solution. If it is just water, then it will freeze, and when water freezes, it make crystals, right? And these crystals can just poke into the cell and burst the cell, and cell dies. So we don't have to do that. But how do we make the cell competent? Now there is one way to do it is by treating the cells with divalent cations like calcium. Okay. Now what does divalent cations like calcium do? They increase the efficiency, or they increase the efficiency with which DNA enters the bacterial pore through its cell wall. Okay. So one way to make bacterial cells. There are multiple ways. We'll talk about all. First is treating with divalent cations, calcium 2+. Okay. So write down, this increases the efficiency of DNA entry into the bacteria. This increases the efficiency of DNA entry into the bacteria through the pores in its cell walls. So there are always minute pores in cell walls and cell membranes of living system. That's how they can, exchange certain important things which they need from the environment. Even our cells have these pores. Okay. We can also do one more thing, which is called heat shock treatment. So you give heat shock to the cells, and in response to heat shock, cells kind of contract and relax, like so their pores becomes bigger. So whenever we heat things, they expand, right? You know that heating leads to expansion, and then freezing back or cooling down leads to contraction, right? That happens with abiotic substances as well, with molecules, etc. Yes or no? And am I making sense, people? When we heat DNA denatures. From its coiled form, it becomes linear and expands and opens up.
Yes, sir.
Same thing. When you give a heat shock treatment, how it is done is, so for a heat shock treatment, what we do, we first keep the bacteria on ice. So ice is around 0 degrees Celsius, right? So we keep it chilled. Then this bacteria which is present on ice for a very long time, then we suddenly put it to 42 degrees Celsius. Now this acts as a heat shock. It is 42 degrees Celsius is enough a difference to give a heat shock, and it is also not so much that it will kill the bacteria, because if you heat it too much, you don't want to boil it and kill it, right? It should not burst. Then we keep it on 42 degrees Celsius for a brief time. Okay, now this is somewhere around 5 to 10 minutes. That's all. Brief heat shock. And then we have to put it back to ice. Back to ice. Okay, and in this time frame, it's enough to give the heat shock, and it makes the bacteria to be able to take up the recombinant DNA. So when we are keeping it on 42 degrees Celsius, we keep it for briefly with the vector, plasmid vector, and that in that moment, many plasmid vectors can just enter, or these bacteria can take up because their pores are bigger now. Make sense? Is the logic clear to all of you? So the reason I focus more on logic is so that when these questions come in your exam, you don't have to remember each and every answer. If you know it, you can just formulate it into sentences and write it, correct? In your own words. Make sense, everyone?
Yeah, sir.
Yeah, cool. So this is another way. The third way is called using a microinjection, but this is not a very high throughput way. That way you cannot do you cannot send plasmid into multiple cells, too many cells. So it is called microinjection, and as you can understand by the term, it's exactly injecting the plasmid directly into the nucleus of a cell. Okay, this we can do for bigger cells like a animal cell, like a lymphocyte or a big WBC of a human, but not like very small cells like bacteria. It's not possible. No, not that much feasible. So write down in microinjection, inject the recombinant DNA, inject recombinant DNA directly into the nucleus of animal cells, or even in the cytoplasm will work because it's a plasmid. The nucleus or cytoplasm of the animal cells. Okay, mostly we do it in the cytoplasm. NCERT says nucleus, but that can also be done. Okay, so this is mostly microinjection is done for animal cells, where we need to just transform a few cells, and then they will keep dividing to make more and more transformants. So there this method can be used. Heat shock is used, and treating with divalent cations is used mostly for bacterial cells. There is one more method which, if I'm not wrong, in the field is mostly used for plant cells, and it's called a gene gun. It's also known as biolistics. It has come from the word ballistics. You know what is ballistic? Ballistics is
like an explosive, essentially.
Yes, basically you are sending an explosive packed in something like bullets and missiles and things like that. So that's ballistics. So we are using a gene gun, and we call it biolistics. So what is a gene gun or biolistics? Is similar to using a gun with bullets. Here we use pathogen vectors, and the cells are bombarded with high velocity microparticles. So we take microparticles of gold or tungsten, because both are inert metals, right? And do not play a role or they do not react with enzymes or play a role in biology. Most like they are inert. So gold and tungsten coated. So these microparticles are coated with our DNA of interest. Okay, coated with DNA particles. So you coat that microparticle with DNA particles, basically your vectors, and then shoot it, like shoot the cells with, bombard it with these gene gun. That's why it's called gene gun. It's not a bullet gun. The bullet is replaced by gene, and then they will just enter into the cell. In this process, they are high velocity, so they just act like a bullet, make a small hole and go inside. But that small hole is not lethal enough to kill the cell, and the cell heal itself, but our gene is inside. Yes, Fatima.
So what about the gold or tungsten? How will it not harm the cell?
Because it's inert metal, they don't do anything inside the cell. They are not major activators of pathways like magnesium, iron, calcium. They can do other physiological things inside the cell. So, but gold and tungsten are mostly inert particles. They do not react, so that's why we use gold or tungsten microparticles. Even in nanotechnology, if you know, the future of medicine is people are trying to administer medicines through nanotechnology, to make medicines more efficient, so that they reach exactly where they. So you want to pack the drugs in nanoparticles made up of nanobeads made up of gold, tungsten, or these metals. They will reach their target organ and then open up and release the drug there only. So your drug, so for example, if you have an infection in liver, but you still eat the medicine through mouth, it goes into your stomach, then absorbed from the intestine, flows everywhere in your blood, and then reaches the liver and acts there, right? That's why all medicines have some side effects, because they also go to other systems where they are not showing their healing properties, but doing something to alter the normal physiology. That's why some medicines elicit vomiting, some medicines have headaches, some medicines elicit. So if we can in a targeted manner give medicine exactly to the tissue which requires it, the side effect part will come down and the medicines will be more effective, and you will have to give less dose. We always give more than what required, because we know that most of it is going to lost here and there in causing some side effect, or will never reach the target organ, because it gets mixed in the blood and stays everywhere in the body. Make sense, everyone?
Yes, sir.
So that's how it does not affect the physiology. Gold or tungsten does not affect the physiology. They might be doing something. I will not say that they do not do anything at all, but they are the most inert or the least reactive. So that's what we can say. Okay, Fatima? Like your concern is exactly correct. Yep. Okay. But then we cannot send that without sending something inside the body, correct?
Yeah.
Yeah. So we have to do a trade-off. So this is called gene gun or biolistics. And there is one more technique where we use pathogen vectors. Okay. So we actually use the vector itself, like the virus. So and that was done exactly in cases of making vaccines, COVID vaccines, where we used adenovirus vector. So we just take the virus and take out its genome, just the capsid, and put our whatever we want to send inside the cell in that vector. And then we rely on the virus's mechanism to just do what it does. It goes in the cell and infects the cell with its genome, but instead of its genome, now it is our recombinant DNA sitting inside its capsid. But that the virus does not know. So it will just do its normal work, go and infect the cell, and when it is infecting the cell, it will transfer our recombinant DNA into the host instead of its own genome. So that method is called using disarmed pathogens as vectors. That's again a product of biotechnology. So we don't have to do much of work. Virus is good at infecting. If we can convert that virus into a vector, and to be safe, we don't use human virus on humans as vectors. So we use something similar like a chimpanzee virus, like a adenovirus, which infects chimpanzees but does not infect humans in general. But when we modify it, tweak it a little, it can still transfer whatever is inside into a human cell and will act as a vector, and then gets degraded because it cannot cause much harm to a human system. So that's also there, but that's not you don't have to study that in detail, so I'm skipping it, but you just know that's another way to send our recombinant DNA inside the host cell. Is that clear? So just remember that for working with bacterial cells, this and this is mostly used. I'm writing back. For animal cell, this is used mostly, and gene gun is mostly used for plants. It's not like it cannot be used for bacteria. So for example, microinjection is very difficult to be used for bacteria, but a microinjection can be used for plant cell, or gene gun can be used for animal cell as well. Gene gun can also be used for bacterial cells, but treating with divalent cation and heat shock is a simple way to make competent cells. So we just do it that way. If you can do something in by using 100 rupees, you would not like to do the same thing using 10,000 rupees in a fancy way, right? So that's the thing. In science, money is a very important factor because it's coming from the taxpayers, you know, like your parents and my parents, they are paying the tax, and that's why, like, so basically I am doing science. The work that I am doing, my salary, my fellowship, everything, my stipend, grants, the money to buy chemicals and do things, that's all coming from the taxpayers' money. So I should use it judicially, and I should also be responsible and answerable to the society for my science. So that's there. Okay, cool. So is that clear? Is every step clear to you now? So
Yes,
all
sir.
the tools I have taught you right from the beginning, where we cut our gene of interest, then take out the plasmid from bacteria, cut the plasmid using the same restriction enzyme. What is a restriction enzyme? Then how to put different, what different components should be there in a plasmid vectors, like ORI, gene of interest, selectable marker, all should be there. Then how to ligate it back to make a recombinant DNA, and how to send it into the host cell. So all of this process now you have to do in a sequential manner, which is the last section of this chapter. And I think if I've noted it down, please just check it if I have noted it down. If you come to page 201, I've just noted down the page number. Please open 201 in your books and tell me, is there a section 11.3 saying processes of DNA technology, something like that? Can anyone just confirm? Am I audible?
Yeah, so there is page 201.
There is. So yes. So Matthew, will you please read the headings for me? Just the headings. So there'll be some 1, 2, 3 in that.
Genetic material, cutting of DNA at specific locations, amplification of gene.
Sorry. First is isolation, right?
Yes, isolation.
Yeah. So I think this you have also done in your schools as practical. We must have
That's
isolated DNA from banana or something, or any fruit, correct?
Yes, sir.
So that's the first process, to isolate the DNA, correct? And then
Cutting of DNA at specific locations.
yes, this you already know how to do it using restriction enzymes. Then and then separating it using the gel agarose.
Then amplification of gene of interest using PCR.
Using PCR. We have not discussed PCR, right, in detail? Yes.
No, not
So
in detail.
yes. So PCR we will discuss. So by PCR we amplify our gene of interest that we have purified from gel agarose. Agarose gel electrophoresis. PCR I will take up in the next class. Then so we just amplify. So we amplified, let's say. Then
insertion of recombinant DNA into the host cell or organism.
yes, this we have done just now. We can use any of these methods to insert it inside the host cell, and also we can check whether our vector is inserted by using a selectable marker, right? This we also did. Then
obtaining the foreign gene product.
yes, obtaining the foreign gene product means that how are you going to get that protein out on a large scale? So for that you need to know that there are bioreactors. So this part where we obtain, this is also known as downstream processing, where we obtain the final gene product. So we call in the field as downstream processing of the gene of interest. So we let that gene of interest get replicated, transcribed, and translated into protein. Then we culture these bacteria into something called bioreactors in very, very large quantities. And then these bacterial cells are then lysed and our protein of interest is purified. So this part I will take up in the next class. Okay, is that clear?
Yes,
so
sir.
before that, on Saturday coming, so which will be your, so next week Saturday will be your test. So I'm announcing it today. I will be taking a test of your genetics because biotechnology includes genetics. So just write down that chapter 5, 6 and 11, 6 and 11, this current chapter, excluding the PCR part and the downstreaming process, but whatever we have discussed, these 3 chapters, testing your genetics, basically these all are talking about DNA, gene manipulation, genetic engineering, etc. So this test will be conducted on Saturday. I think Saturday is what date? 26th. And I want all of you to please give this test because this will be an idea for me to evaluate where you lie in terms of knowledge.